Hello students, today we will see the next chapter, that is protein. In the previous lecture we have seen the last topic regarding to the lipids, that is the characterization and analysis of lipids. Now today we will see introduction to the proteins, what is the importance of proteins, then contents which are present in the proteins, and the last one, that is the Jadhav method for the estimation of proteins. Let us see one by one. So the first one, introduction. So the proteins are an abundant component in all cells and almost all except storage proteins, and it is very important for biological functions and cell structure. Food proteins are very complex one. So the many food proteins have purified and characterized. Proteins vary in the molecular mass ranging from approximately 5,000 to some millions of Daltons. Proteins are composed of elements including hydrogen, carbon, nitrogen, oxygen and sulfur. 20 alpha amino acids are the building blocks of the proteins. The amino acid residues in a protein are linked by peptide bonds. Nitrogen is the most distinguishing and abundant element present in the protein. However, the nitrogen content in the various food proteins ranges from 13.4 to 19.1 due to the variation in the specific amino acid composition of the proteins. Proteins can be classified by their composition, structure and biological function or solubility properties. For example, if you consider the simple proteins which contains only amino acids upon hydrolysis, but conjugated proteins also contain non-amino acid components. Proteins have a unique conformation that could be altered by denaturants such as heat, alkali, acid, urea, guanidine, organic solvents and detergents. So generally the denaturation, it is the process in which the protein loses their quaternary, tertiary or secondary character or structure present in the native state. Now next is the importance of the analysis. So the first one, nutritional labeling. So if you consider any kind of food product is concerned, so that there should be the nutritional labeling. So what is the value of the protein present in the given sample? So for that purpose the analysis of protein is very important. Then next one is the pricing. So the cost of certain commodities is based on the protein content as measured by the nitrogen content. For example, if you consider cereal, grains, milk for making certain dairy products, for example cheese. So for that purpose, depending upon the particular composition, the pricing of the particular food product is decided. Then next one is the functional property investigation. Generally, proteins in a various types of foods have unique food functional properties. For example, if gliadin and glutenins in the wheat flour for bread making, casein in milk for coagulation into cheese products and egg albumin for foaming are the various functional property which are having importance. Then the next one, biological activity determination. So some proteins including enzymes or enzyme inhibitors are relevant to the food science and nutrition. For example, the proteolytic enzymes in the tenderization of meats, pectinases in the ripening of the fruits and trypsin inhibitors in the legume seeds are the proteins. To compare between the sample, enzyme activity often is expressed in terms of specific kind of activities, meaning units of the enzyme activity per mg of the protein. While protein analysis is required when we have to know that the total protein content, again the content of a specific protein in the mixture, then protein content during the isolation and purification of the protein, then what is the non-protein content present in the sample in terms of the nitrogen. Suppose nitrogen is present in the form of proteins also and non-proteins also. So by analyzing the sample of protein, we can know that what kind of nitrogen is present in the sample. Also we have to find out the amino acid composition, what kind of which amino acids are present in the sample, we have to find out from this. Then nutritional value of the protein. So by using this we have to find out the nutritional value of the protein. Then next, what are the contents of the proteins in the food? So if you consider various categories of the food items, then we can say that if you consider first cereals and pasta, so from that the wheat flour and whole grain, which consisting of the highest percentage of the protein, while the cornstarch having lowest percentage of the protein while considering the cereals and pasta. While if you consider the dairy products, so from the dairy products, the milk, non-fat, dry, having highest percentage of the proteins, while that of milk which is having reduced fat or fluid 2%, which is having lowest percent as we concern about the dairy product. If you consider fruit and vegetables, so the apples having lowest percentage of the proteins, while asparagus having higher percentage of the proteins. If you consider again potato, whole potato, it is having also 2% of the protein. If you consider leguminous plants, particular soybeans seeds having highest percentage of, that is 36.5 percentage of the protein, while tofu having lowest percentage if you concern about the legumes. Then meats and poultry fish products if you consider, then beef having highest percentage of the protein, while the raw egg having lowest percentage of the protein. Now next one is the different methods. So generally principles, general procedures and applications are described for various protein determination methods, such as generally for the estimation of proteins we have used Jadhav method, Dumas method, and third one is the infrared spectroscopy method. Beyond that, other methods are commonly used in nutritional labeling and quality control applications. Now the next one is the Jadhav method. So what is Jadhav method? So the Jadhav method, it is the method which is useful for the estimation of nitrogen from the given sample. So this method is also used here because nitrogen is the one of the constituent present in the protein. That's why it is used. In the Jadhav procedure, proteins and other organic food components in a sample are digested with sulfuric acid in the presence of catalyst. Some kind of catalyst are there and there is a decomposition or degradation of organic compound is going to take place. So the total organic nitrogen is converted into ammonium sulfate and that digest is neutralized with alkali and distilled into the boric acid solution. So the borate anions formed are titrated with standardized acid which is converted to nitrogen in the sample, and as the result of analysis represents the crude protein content of the food, since nitrogen also comes from the non-protein components. So as we know that whenever we have analyzing nitrogen from the Jadhav method, whatever be the source, it is either protein source or non-protein source, the nitrogen is comes. That's why this is one of the disadvantage of this method, that we cannot analyze individually the protein. Nitrogen comes from the protein. Now this is the instrumentation this is the particular apparatus which shows the particular, this is the Jadhav flask where we have to take a sample along with catalyst and NaOH. Here we have first performed the digestion of this particular sample where the organic nitrogen is converted into ammonium sulfate, and then after heating it should be condensed into the standard acid solution of known volume. So we will see one by one. So first we have to perform the sample preparation. So in order to prepare a sample, solid foods are ground into the fine particles, that is about we have to take a 20 mesh screen and passing via the 20 mesh screen, okay, and then samples for the analysis should be homogeneous, okay. So no any special preparation for the sample is required, only these kind of things are required. Now the next step is the digestion. So for the digestion we have to accurately weigh the sample and place the sample into the digestion flask. Then into that we have add acid and the catalyst. Then we have to digest the given solution under heating until we have to get a clear complete breakdown of the all organic matter. So the non-volatile ammonium sulfate is formed from the reaction of nitrogen and sulfuric acid. So reaction is what? Protein whenever reacts with sulfuric acid under heating, it will give rise to the ammonium sulfate. So during digestion, protein nitrogen is liberated to form the ammonium ions. Sulfuric acid oxidizes organic matter and combines with the ammonium form, and that is nothing but the ammonium sulfate. So the carbon and hydrogen elements are converted into its corresponding oxide, that is the CO2 and H2O. Now the next step is the neutralization and distillation. So the digest is diluted with water, okay. So the alkali containing sodium thiosulfate is added to neutralize the sulfuric acid. The ammonia formed is distilled into the boric acid solution containing the indicators, that is mixed indicator methylene blue and methyl red. So that sodium sulfate and NaOH mixture is there, okay. It will liberates ammonia, and along with that sodium sulfate is formed along with water. So that ammonia is liberated, is reacted with boric acid, it will form a ammonium ion, along with there is a formation of borate ion. This particular borate ion when reacts with standard acid, okay, it will form a boric acid, and the borate ion are formed which is proportional to the amount of nitrogen and which is titrated against boric acid, and in this way we have to find out the amount of nitrogen present in the given sample. So the calculation, now the number of moles of HCl which is exactly equivalent to number of moles of ammonia which is formed, which is equals to the number of moles of nitrogen in the sample. So a reagent blank should be run to subtract the reagent nitrogen from the sample nitrogen. So the percentage of nitrogen is equals to normality of HCl into corrected acid volume upon weight of the sample in grams into 14 gram into normality 14 gram into N upon mole into 100. So where N is the normality of HCl, corrected acid volume is equals to ml of standard acid for a sample minus ml of standard acid for a blank. 14 it is the atomic weight of the nitrogen. A factor is used to convert percent nitrogen to percent crude nitrogen. So what is the need for the convert that percent nitrogen into percent crude nitrogen? As we know that the particular sample containing non-protein nitrogen also there. That's why we have to convert that percent nitrogen to percent crude nitrogen. So for that purpose most protein contain 16% nitrogen. So the conversion factor is 6.25. Okay. So that percentage of nitrogen upon 0.16 it is equals to percentage of the protein which is present in the given sample, or we can write it as percentage of nitrogen into 6.25 equals to percentage of the protein. Now what are the advantages of this method? So this method is applicable to all types of the food. Then this method is very inexpensive. Then this method is accurate and official method for the crude protein content estimation. So it has been modified, that is a micro-Jadhav method, to measure the microgram quantities of the proteins. Then disadvantages. Generally this method measures the total organic nitrogen, not just the protein nitrogen. We have already seen that it measures the crude nitrogen. So the method, it is somewhat time consuming. It requires around 2 hours to complete, and we require corrosive reagents for the analysis or estimation. So this is all about the Jadhav's method. Okay. So in the next lecture we will see the other methods such as the Dumas method and IR spectroscopic method. Okay. Thank you.